still in the chat, really with some practical tips and so questions for some practical advices. And people really would love to see your live demonstration video, so maybe we can think about how we can make that possible. But for now, we are moving to the next tutorial, to the next presentation, and we will have a small swap in the program. So first we will go with Lipid Data Analyzer, and then we will go to the Lipid Finder afterwards. So I'm happy now to introduce Juergen Hartler. And Juergen, please share your screen.
Thank you very much for this kind introduction, Maria. Do you hear me?
Yes, perfectly.
And can you see my screen?
As well.
Okay, perfect. Good. Thank you very much. Then let's go. So I say a big thank you to the organizers, to Maria and Valerie, that you invited me to give a talk at this exciting event. And it's great to see that so many people want to watch a tutorial on LDA. Thank you very much for attending. At this point, I want to mention that I won't provide, that I will provide more a training session on the LDA, because I had been asked that I should do this for non-experts. And my aim of this talk is that you can use the tool afterwards and you will get out the best of your data. So first, in the first line, I wanted to prepare this whole thing as a hands-on session. Sorry, I didn't want to do that. I want to have the pointer. And so everything that you see now here, you can find on this Spring School page here. There you have installation instruction of the LDA. Then here you will have installers. And here's as well a data description, and you can download exactly the test data that I will show you in this tutorial. And I will show you now how principally data analysis with LDA looks like. First, you need your MS data for sure. Then you need the parameters for the 3D algorithm and fragmentation rules. The fragmentation rules make the spectral annotations, and then you need mass lists to tell the LDA what it should look for. Then the LDA will do automatically the quantitation. Then you can have part of the manual correction, and I will show you I will spend most of the time in this part so that you really get rid of false positives. Then you have the statistical analysis section, and then you can export your results to different file formats. I will walk through now all of these parts of the LDA so that you can make a complete data analysis of your data. The first thing I will start with is the MS data. The LDA can use MS data directly, the raw data of those vendors. There is an it integrated the MS convert, but this is only available for the Windows installation because we have here integrated DLLs and they are only available on the Windows. And then MS Convert will convert the raw data to the MC-XML format, which is the primary input format of the LDA. And this one can then be used for all platforms. For Waters, you have to make a separate installation of Mass++, and then you have to change a parameter in the settings file, and then you can use as well directly the Waters data. The LDA will then convert the MC-XML to the Chrome file format, which is an LDA internal format for efficient data access. So we have now the part of the MS data. The next one is the parameters for the 3D algorithm and the fragmentation rules. This is more or less the setup of the LDA. The only thing what you have to do is start the LDA, go to this settings tab. Here you see the current settings, and then select which MS instrument you have. And this is for the 3D parameter, for the MS1 quantification, the 3D parameters. And then you have to select the collision energy of your instrument, plus and minus. This will load the fragmentation rules. These fragmentation rule sets have been developed on authentic standards, and they are show a very high positive predictive value. And as soon as you have selected one for the positive and one for the negative ion mode, then simply click on Save as Default, and then you have set up your LDA. Every time you restart the application, this will change, and every time you restart your application, you will have this setup for your machine. So the next thing is then the mask lists that you need to provide. The LDA comes with several mask lists. You find them in the installation directory of the LDA. I installed here LDA under Program Files LDA, and you find there a folder called Example Mask Lists. And the most comprehensive ones are these ones here, the negative and the positive XLSX. And here I have to mention the extension for the sphingolipids that Ed mentioned yesterday. The negative and the positive are designed for the platforms where we already made this extension. This is currently the Velos Pro and part of the Q Executive. And for the other platforms, please use this one, negative underscore no sphingor OH, or you don't see it here, positive underscore no sphingor OH. I will show you in this slide what is the difference between both of them. So this is the way that these mask lists look like. This is a simple Excel file that have here at the tabs are the lipid classes that you want to search. If you don't want specific classes, you simply delete the tab. Then you have to provide a name, arbitrary name, and if in the case it's a lipid, it should end by the number of carbon atoms. Then the number of double bonds, and then you have the chemical formula in this tabular format. And the next thing is you have to provide the masses of the adducts. And as I already mentioned, we provide these mass lists for many lipid classes. And this is an optional parameter, which is quite useful if you have, for example, MS1 data only. You can specify here the retention time where you expect the lipid to be detected. But for most of the data, you don't need this, because when you have MS2 information, the LDA can differentiate between different isobaric and isomeric lipid classes. But if you have low resolution or only MS1 data, this is quite useful. And then you can tell the LDA here what is the retention time range for this class. And this is as well an optional parameter. You don't need to do this. And the one difference with the new extension is that you have to provide for the sphingolipids this OH number. That means you have this mass list only for a single, for example, the dihydroxylated version. And then you say tell the LDA for which hydroxylation range it has to search for a species. And here it would go from mono to trihydroxylated species. So this is all required. This is required for starting the quantitation. For the quantitation, you have an graphical user interface. It's the first step here, quantitation, but this is only for a single file. What you have to do is to specify your raw data in the CROM format or the MCXML or your raw data directly. And then in the second line, you have to specify the mass list in Excel format. But this would be quite tedious if you have to do this for each and every file. So therefore, we have the second tab here, which is for the batch quantitation. And here you specify the directory where your raw data is, which contains your raw data. And then you specify the directory where your quant files are located. And then you have there some additional parameters. This is the time tolerance before and after when you specified a retention time in the Excel file. If you didn't, you don't need to mind about this parameter. This one is a threshold in relation to the strongest peak. It's mainly, I would say, more to accelerate the algorithm that you remove the very small ones. And this one here is a retention time shift parameter. So when you, for example, you specified your retention times in the Excel file, and then you see suddenly a shift, then you only have to put here the value, how big the shift is, and this will be automatically applied to your mass list file so that you don't have to redo this all of the time. Then these two parameters are the following. The LDA calculates the monoisotopic peak, and with this parameter, you can say how many additional isotopes it should use for the quantitation. Typically, we use 2. And this, the LDA checks additionally if the isotopic intensity distribution matches the really identified isotopes. So this is for removing false positives. I would recommend that you don't change these two parameters. They work quite well for lipids. And here, the last parameter down here is how many processors do you want to use for a quantitation. The LDA reads out how many threads you have available and typically reduces by 1 so that you still can continue working. And I would recommend that you use it in this way. Then you simply click on the Start Quantitation button, and the LDA will start with that. Here, a green check mark will say, okay, the quantitation has been finished. Red arrow and X will say, okay, there was something wrong. And here on the right side, you see the progress of your quantitation. When this is done, you will see this is the first data set, the standards that I provide on the mass list. And this contains here as well this directory. And if you use both of them, you will get those results out. These are the LDA results files. In fact, they are Excel files. I don't want to go into details how these files look like because the LDA provides sufficient methods to access these files and to export them and so on. And I will go now to the next section. This pertains to the data analysis. You have there the manual correction, which you can do, which is not a must, and then you can go to the statistical analysis and export it. I talk about those two sections together. The reason is because they are quite linked to one another, and I will show you now why. The easiest way to visualize your results is that you go to this display results. Here you can give the MS data in the Chrome format, and the second one is then the results in Excel format that the LDA produced. But this would be quite tedious if you do a manual correction on each and every single file. So the typical way to do the manual curation of your data is that you use the statistical analysis section. Here you click on the Add Directory. Then you give the directory where your files are located. I take now here the samples of the second data set. These are the hepatocytes zip directory. You simply specify this directory and click on Open. And then it will automatically load all of the Excel files that are in this directory. If you have some additional Excel files in it that were not produced by LDA, you simply can select them and remove them. The next thing is that you have to specify retention time, grouping time. This one is an optional parameter so that you only can be sure that the species will appear exactly in the sequence of your mass list. So it's recommended that you select here the mass list that you use for the quantitation. And here there is an additional parameter that you can put in cutoff settings for each and every lipid class. This is works a little bit different. First of all, it selects as a reference the strongest lipid species of each class, for example, phosphatidylserines. And the second advantage of this filter is it does not blindly remove you all of the species. It shows if a single species exceeds the thresholds given here, all the others identification of these species are still kept. The reason is so that you don't produce any missing values for your statistics. But if all of the species are below this threshold value, they will be removed from the next analysis step. Then you click here on the Accept button. And what will happen now, before we were in this selection tab, now you will here the tabs will pop up for each and every lipid species that you have in your results files, and heat maps will be generated for those species, and those are interactive. You have here at the columns the names of your MS runs. Here you have the names of the species, which is in fact the bulk nomenclature underscore the retention time. And at the bottom, you will have an export panel to the different file formats. And here you will have a settings panel. The relative intensities are color encoded. Here you will almost everything is black. The reason is because this is a technical replicate of the same liver sample. And what does these colors here mean? Gray means the species is not detected. If you have a yellow border around one of the species, it says that there has been more than one peak identified. This typically means that you have to pay attention to this species because maybe one of them is wrong. Then blue means that some adducts are missing. This is typical for species of lower abundance. The higher ones should have all the adducts. And purple is a combination of both of them. So you have more than one peak and some adducts are missing. And if you hover your mouse over one of these rectangles here, this will show, you will see it in white. And for example, I will show you now how this interactive heat map works. You hover the mouse over the species, then you will see here at the bottom the name of the lipid, at which retention time it was detected, at which sample, and here the standardized area and the quantified area. In this sample, I didn't put in any standards, so both areas are the same. When you then click with the left mouse button, it will automatically jump to the section where you can verify your data. So what I showed before in this display results, it jumps automatically over here. For example, we have here the PC44 colon 0. And what you will see here is immediately a 3D viewer of your species. And here, these are the small ones, the red ones. And here you see the chromatogram. What can you now do with this MS1 viewer? You have here the selection of your lipid classes. And here you have your detected species with the name, retention time, and adduct. And here in the 3D viewer, you can see in the default view the monoisotopic peak, the first isotopic peak, and the +2 isotopic peak. And the same you have here in the chromatogram viewer. You have here only this slice. And if on the left side and the right side, you have zoom settings for your 3D viewer and for your chromatogram viewer. And what I typically recommend to do is to check if the peak integration works well. The LDA comes with many 3D parameters, but it's good if you zoom quite close. And what you see here is that everything that is red has been used for integration. As you see, the LDA fits here an ellipse around the peak, and what is green has not been used. So if big slices of the peak have been cut away and are not red, then there is something wrong. So I recommend when you first use your data sets, try this out for very small species, a very small peak, a very strong peak, and then for a very low MC value and a very high MC value. If all of them are quite well integrated, you are ready to go. If not, then you can shoot me an email and I can try to help you with that. So then the next thing is you want to verify your spectral annotation. For this, you only have what I forgot to mention is the species that is currently selected is here color encoded in blue. You simply click the right mouse button on the species, and then you go down here to show MS. And then you will this view here will change, and you can see here the spectra viewer. Black means that this fragment has not been annotated. Brown, that's a head group fragment, and all the other colors mean that those are chain fragments. Then you will have here on the right side information about the spectrum. The first line is that it's for this precursor spectrum number 10 out of 13, the retention time, the precursor, and at which MS level it's recorded. So if you have MS3 here, it would stand 3. And then here you can scroll through the spectra. With plus, you go to the next spectrum, with minus the previous. And here you have the chromatogram. There is a small blue line, and this shows you the current location. If you're getting out of the peak, you won't have any annotations any longer. And here you have, again, zoom settings. I will now zoom into this particular part here down here so that you can see the chain fragments. And you can see now here quite nicely the chain fragments of the protonated BC species. And you see they are quite low abundance. It's around 1 to 3%. So what do now these colors on the left menu mean? White means that there is no MSN information present. So this peak has been, this species has been identified by MS1 information only. Green means that this spectra had been unambiguously assigned to this lipid species. Orange means that you have to pay attention. The LDA algorithm could not discriminate between two or more species. You see both of them are at the same retention time and they will have the same precursor mass. And yellow means the same, but here the LDA takes a look at the MS2 spectra, and here it found a gradient in the spectra. That means one part of the peak looks like PC46, the protonated version. This one looks rather like the sodiated version of 38.3. And when we take a look at this spectrum, we can see here this is the location of the spectrum at 26:43 minutes, and you see here quite nicely the fragments of the PC. But if you go one spectrum further, you see that these head group fragments are quite high, and these are typical for sodiated PC species. So the LDA recognized that this peak is an overlap of two species. And what it does here, if it finds such a gradient, it splits the peak between the two spectra, which are different. So it makes here in the middle a split. And if you take a look at it at the MS1 level, you can see that the right part has been used for the quantification of the sodiated species, and the left part for the protonated species. But you have further co-eluting, not only from different lipid classes, you have as well co-elution of chain isomers. For example, we have here PI, phosphatidylinositol, 36:3. And if we take a look at the spectrum, you can see that this peak consists of two species. The one is a PI18:1, the other one is a PI18:1/18:2, and the other one is a 16:0/23. And here the LDA does the following. It takes the quantity of the MS1 peak, and then it splits according to the distinct fragments, the intensity between the two co-eluting species. So now let's go to the manual curation. What is very important if you're doing a reverse phase is the retention time. For example, you can see here a series of identified triglycerides that has been published in this publication from Harald Köfler, from Harald Köfler's group. And for example, you see here the 46:1, and if you go down, the 48:1 elutes a little bit later. So that means increasing number of carbon atoms then the species elutes a little bit later. When you have an increasing number of double bonds, it elutes a little bit earlier the species, and it decreases the retention time. And this was then later on called the equivalent carbon number model by the group of Michael Holzschabeck. And what we did is we make use of this model. The LDA uses the following formula. This is the retention time is dependent on the number of carbon atoms and double bonds. And then we found that this formula fits quite well for different chromatographies. And what the LDA does, it takes only the green heats, the MS2 spectra, which are conferred by MS2 spectra, and then it fits this model by a Levenberg-Marquardt algorithm. And then you have the parameters. It doesn't matter which reverse phase chromatography are you using. This works principally quite well, and makes then a prediction so that it says for the ones where we don't have MS2 spectra, where should those molecules be? And we used this formula because the dependency on the number of carbon atoms looks like a logarithmic function, and the one for the retention time influence of the double bonds more like an exponential decay. And so what does this help you now? You see here, for example, the results for phosphatidylinositol. We have here the 44:1, then the 44:2 should elute a little bit sooner. This one it does, the 44:3 as well. But if you, for example, look at the 44:0, this is completely wrong. And this one here as well. So the LDA automatically uses then this retention time prediction and removes everything that is too far off from the predicted retention time. And you see that you will end up with a with a much more neater list in the end. And you can see that the false positives are gone, and you can still make use of the ones where you didn't identify MS2 spectra, but you can still be quite confident that this is the correct molecule, because the retention time coincides with what you would expect. So then you have several options for manual curation down here. And when you now want to, for example, manually quantify a peak, the only thing you have to do, you have to click with the right mouse button in the peak area, and then a pop-up menu will appear where you can select the different options of peak quantification. And the typical one is the 3D algorithm, but the LDA has as well some 2D algorithms. In this example, this peak here is the second isotopic peak of the 34:2 species, and with the 3D algorithm it won't work. But if you still want to quantify it, then you have to use one of the 2D algorithms. And here I switched off the texture so that you can easily see the difference. Here the 3D algorithm is ellipse, and with a 2D algorithm you have a certain slice where you extract the chromatogram, and you only take the peak borders this direction, and so it's much more easier that you, if peaks are closely eluding, that you take intensities of another peak. So, but this manual curation would take a lot of time, so we implemented some easier means to do that. For example, if you need to eliminate double peaks, so the first thing you have to do, you go to a single species, to a single run, and say, okay, this MS time is the correct one. You delete the wrong one. Then you have to only go back to the heat map, hover the mouse over the species, and say, click the right mouse button and say, choose analyte just one peak for doubles. This will then remove, it will ask you then, do you really want to do this? This is the species, this is the retention time. And you can select the adducts. And if you click OK, all of them, the yellow ones are gone. The wrong double peaks were eliminated. The same you can do, for example, for missed hits. The LDA is quite stringent with the isotopic pattern, and if you have, for example, a very low abundant hit where you have only the monoisotopic peaks, then you can do this. These two have not been identified. These are in gray. You simply go quantify one, at least one, if it's not already present. Then you hover the mouse over the MS run and species and click with the right mouse button and say quant analyte at not found. And then you have again a selection, which adducts do you want to use? Then you click the OK button and you will see the 32 are identified. Sometimes if you really have no signal at all, then they still will remain gray. To remove hits, you simply go with the mouse over the species. You click with the right mouse button, select analyte. You can select several ones that are then shown in blue. And then you say remove analyte in all probes. And now you again ask for the adducts. You click on the OK button. And the 46 have been removed. So, and there is one other option how you can verify if this everything is correct. This is called Chrome export. You can select here species. I selected here 32, 0, 1, and 2, and here you have the different MS rounds, and in this view you can see all of them come at the same retention time. So this looks quite good that you don't have a deviation. And if you click then here on the export options, you can change the column and the rows. And for example, you can check here if the illusion profile, if the 32.0 and then the 32.1 comes a little bit sooner, and the 32.2 comes a little bit sooner as well. So then a very important thing are the decision rule sets. They do your spectral annotation. In fact, this is a simple language how you can specify fragments. you simply specify an arbitrary name of a chemical formula, then the charge, and at which MS level it's observed, and it has some placeholders. And what I want to mention is that it searches for all possible chain combinations in this process. If you have two chains, it tries out all chain combinations that match to the total number of carbons and double bonds. And these names you can then use in intensity rules, which are in fact should reflect the natural fragmentation pattern of this lipid species. And the whole decision rule set would look like that. So you have a head fragment. That means you verify the head group information. You have the fragment, and you have the intensity rules. The same you have for the chain section, fragment rules and intensity rules. And for the positions, you have only intensity rules. So we provide you with more than 1,000 rules in the LDA that we developed on authentic standards. The benefits of those rules are that one decision rule set replaces thousands of reference spectra. For example, we compared to Lipid BLAST, and there a single TG rule set replaced more than 10,000 spectra. It tries out all possible chain permutations. It's easily extensible. Recently, we have published this work. This did a collaboration partner in Vienna. Here, for example, it's extended the LDA to GIPC. It avoids structural overinterpretation because when the LDA verifies the head group fragments, but it can't find any chain fragments, it doesn't report any chain fragments. It stops at the structural level it could really verify. And it's robust for information from noisy spectra. Here it's identified a diglyceride, even if there was so much noise around. And it returns quite reliable results. We benchmarked here the LDA in 2 studies. This one was about glycerolipids and phospholipids, and here was the extension to the sphingolipid that Ed Dennis mentioned yesterday. And but most of the people are not so familiar with editing text files for fragmentation, so we implemented a graphical user interface to do that. So when you here go to your species and click on Edit MSN rule, you will get an interface where you can enter those rules, and the annotation you can do directly on your real spectra. Here you will see a small video where I changed the formula for the ketene. You will see then here in the real spectrum that this will be removed, and as soon as I correct the formula again, you will see that it appears. So start the video. You can see now here I show with the cursor, with the crosshair cursor. This one is, and now I remove it. You see it's gone. And now I enter again the 2, and they pop up again. So you can easily try to develop your own. Oh, I'm sorry about that. You can easily try to develop your own rules. You have a graphical user interface, and you can do this for any additional lipid classes, and many of the users of the LDA do that. So now let's get to the next part. It's the statistical analysis. I only showed for the part of the manual curation. Now I will take the third data set. It is the one of the triglycerides, what you can do in this section. Here I simply selected the first 3 because they are triplicates of a single group. Then you click on the add group. You give an arbitrary name. And I did this for all of the 3 groups because they are triplicates of different feeding regimes of mice. And then you will have your groups added down at the bottom. For this data set, please unselect this checkbox. You can provide again a mass list for the sequence of the species. I used one that comes with the LDA. It's in the example mass list positive XLSX. And then here in this data set, we added standards. You can provide here the prefixes for your standards. IS was the internal standard and XIS was the external standard. And when you enter this information, you can click on Add Absolute Settings. Then this one will pop up, and you will have an input page where you can type in what were the concentrations of your standards. So that you don't have to do this, I added a file where the standards are already, these values are already stored. Go to load settings and go to the directory where this TG data, which contains the TG data, and select this tag settings.wks.xml, and it will load those species. And if you click now on the accept button, you will have a slightly different view. You have here the heat map, and additionally, you have here a heat map of the group, which looks like that. And when you go here now to the settings, you have here you can select here between different values that the LDA calculates you automatically out of the standard values that you added. For example, I selected here the concentration of the sample volume. Then you can select here the standardization method which you want to use. For the sake of time, I don't explain everything in detail here. And it will change now here to view the following. You hover the mouse now, for example, of this TG561, and you will get now here more information. For example, the concentration of the sample volume, then the standardized area is different than the quantified area, and so on. Then if you click here on a group, you will get a quick overview of your species of this triglycerides. You can easily see which are the strongest ones. When you click here on the species, you will get the bar charts. When you click here on the combined chart, then you can select specific species. I took here the 54 carbon series, and you will get as well bar charts where you can change here the quantities and so on. And yeah, now I'm finished with manual correction and the statistical analysis. And now I will continue with the last section of my talk. This is the results export. You will have export functionalities in the chromatogram view in the bar chart, but the major location where you do the export is at the bottom of the heat map. You can specify here different export options. I show here the ones for the bar charts. There's a little for the group heat map. There's a little bit more like deviation values and so on. Then you have this one is important. You can specify at which structure level you want to export. The species level is the bulk nomenclature. The chain level shows you the fatty acid chains. And if you want to export the position level, you have to click this one down here. Here you can select and deselect species. So you do not delete them completely from your file, but you simply deselect it for the export. Then you have here tab delimited formats like Excel or tab delimited text format, which looks like this. This RDB format is a tab delimited format as well, but it contains more information. So if you don't want to do the manual curation in the LDA and you want to have your own routines for processing this data, then I would recommend that you use the RDB format. And this one here is more for publication and communication. This is a standardized community format, the MC tab format. Here you have this format contains a metadata table, which contains information about the experiment. Then the small molecule table is more or less an overview and the final quantities. Then the features table contains the features, that means the peak borders and the intensities. And the last small molecule evidence table contains the spectra and the ions. So then I am finished with my introduction. What I want to say to you is the LDA is free of charge. There is a separate download page. In the spring school, there is a license until the end of May. The only thing is why we ask you to fill out a license form is that it's for statistical purposes, so anybody can get it free. But just for trying out, I put you a license in until the end of May. Not much. Then it's open source. You can find the open source project over here. We have several published data sets where you can play around with them before you start with your own data. The LDA has a quite comprehensive documentation where you can find more details. I could not cover in this talk all of the details, and this were not the specifics about the sphingolipid extensions. And here you find there are many more publications, but those have to do directly with the algorithms and how the LDA works. And with this, I want to thank all the people that have been involved in this project. I want to thank the organizers that I could give this talk at this excellent session. And I thank you for your attention. And I'm ready to take your questions.
Thank you very much, Juergen. That was really detailed tutorial, how to go step from step. And I guess it was very, very useful for many of us here. And we have several questions, but again, because of the time constraint, we will not be able to ask all of those. But so we can make few live, and then maybe you can answer the rest in the chart. Yeah, maybe some of them actually got answered during the course of your presentation. So
okay,
I will
good.
just take something here from the end. Oksana Rogova wanted to ask something. Oksana, are you here?
Yes, I'm here. Yeah, thank you for a really nice presentation. It looks really interesting. I was wondering, is it possible to do normalization for drift and signal, like based on QC samples, for example, or on the internal standards?
For drift and signal,
yeah.
What the LDA does, in principle, it automatically calculates the retention time prediction values for each and every sample, but it has not anything that does signal drift. What we had, for example, is that we observed in one run that we suddenly had a shift of 8 minutes, but the identification still worked.
I mean, like when we do integration, like total ion chromatogram signal, maybe like it sometimes goes like a little bit up or down. Is it possible to correct for this?
Yeah. I mean, you have the internal standards that you add.
Yeah.
Do you mean those? Yeah. This is possible. Yeah. You can add an internal standard. You simply put it at the end of the mass list. You call it IS something, for example, deuterated standard, and then you can do with the this. I'm sorry, I was a little bit fast in the presentation. In these triglyceride samples, we added there the standards, and this does the signal correction. Yeah.
Oh, great! Thanks a lot.
Thank you.
Maybe there is a question from Alpesh here also with the LC signals. Maybe to follow up on that, Alpesh, would you like to ask a question about the handle rider peaks?
It was really nice presentation, very informative as well. Just wanted to ask, how do we manually curate the peak if there are soldering and rider peaks, co-eluting within this at a similar retention time? How does the software has that functionality to resolve that?
You can typically the function as the LDA should detect them. Because this 3D algorithm looks in both directions, in the MC and the time direction. And in my typical scientific talks, I show that it's possible to distinguish really quite closely eluting species. And if this doesn't work, you can still do it with the in the chromatogram figure that you specify the peak borders yourself. And then there is a function in the heat map again that you say, okay, take the exact peak borders for several samples so that you have the same. But I typically don't recommend this. It's better when the 3D algorithm detects them correctly, and to do an optimization of the 3D parameters that really can distinguish between those species.
Okay. Thank you very much.
Thank you. I hope, was your question answered?
Yeah,
okay.
Thank you.
Thank you.
Great. Okay, so just again, for the interest of time, we will move on, but please, Juergen, could you please go through the chart and maybe comment on the other questions which we have there. Thank you very much again, and also thanks a lot for stepping in